What if you could rewrite the code of life? That's exactly what genetic engineering allows us to do. It's the process of altering the DNA of an organism to change its traits. From disease-resistant crops to potential cures for genetic disorders, this technology is reshaping biology. But how does it actually work? Let's dive into the fascinating world of gene editing and discover how scientists manipulate life at its most fundamental level. At the heart of genetic engineering is DNA, the instruction manual for all living things. Scientists first identify the gene they want to modify, remove, or insert. This gene is then isolated using enzymes that act like molecular scissors. Once isolated, it can be inserted into the DNA of another organism. This process allows for precise changes, whether it's making bacteria produce insulin or creating drought-resistant plants. One of the most revolutionary tools in genetic engineering is CRISPR-Cas9. Think of it as a GPS-guided scalpel for DNA. CRISPR allows scientists to target specific genes with incredible accuracy. It uses a guide RNA to find the exact DNA sequence, and the Cas9 enzyme cuts it. This enables scientists to delete, replace, or add genes with unprecedented precision, making gene editing faster, cheaper, and more accessible than ever before. Genetic engineering isn't just for labs, it's already in your life. Many crops like corn and soybeans are genetically modified to resist pests or tolerate herbicides. This reduces the need for chemical sprays and increases yield. In medicine, genetically engineered bacteria produce insulin for diabetics. Even gene therapies are being developed to treat inherited diseases. The applications are vast, and they're growing every year as the technology improves. But with great power comes great responsibility. Genetic engineering raises ethical questions. Should we edit human embryos? What if gene editing is used for cosmetic enhancements or intelligence? There's also concern about unintended consequences, like modified genes spreading in the wild. That's why regulations and public debate are crucial. As the technology advances, society must decide how far we're willing to go in rewriting the blueprint of life. Despite the concerns, the potential of genetic engineering is enormous. Imagine curing genetic diseases before birth, growing organs for transplant, or engineering crops that thrive in extreme climates. These aren't sci-fi dreams, they're real possibilities. The key is responsible innovation. With careful research, regulation, and public input, genetic engineering could solve some of humanity's biggest challenges. The future of biology is being written, gene by gene. So, what's next? As tools like CRISPR evolve, we'll see even more precise and powerful ways to edit genes. Scientists are exploring gene drives to control mosquito populations and synthetic biology to create entirely new organisms. The line between natural and engineered life is blurring. Whether that excites or scares you, one thing's clear: genetic engineering is here to stay, and it's rewriting what it means to be alive.